No fruit around the world is more famous for the juxtaposition it places on one's senses than the durian. While some claim durian to have a sweet aroma, most others claim its scent to be as revolting as raw sewage and rotten onions. Compare that to its sweet taste and custardy texture, durian seems all the more peculiar. But what is it about durian that causes such a distinctive assault on everyone's senses? Grown throughout Southeast Asia, the durian is ubiquitous to any Asian street market, corner store, or frozen section of a convenience store. Often referred to by its people as the king of fruits, for its strong thorny husk, sweet custardy pulp, and of course, its strong odour. While mainly consumed fresh, it can be had fermented, made into chips, or flavoured foods such as ice cream, yogurt, milkshakes, and curry. Along with its multitude of uses, durian's economic value is nothing to be scoffed at. Thailand, the number one exporter of durian, exported $1.47 billion worth of durian in 2019. This was followed by Hong Kong with $455.62 million and Vietnam with $272.66 million. On the other end, China, the largest importer of durian, imported $963 million worth or 358,000 tons of durian in the first 6 months of 2019 alone. With a large economical influence and distinctive cultural and social impact durian has on the world, it comes as no surprise that a team of researchers, financial donors, and durian lovers have come together to find out what causes that famous stench. In 2017, a team of Singaporean cancer researchers from the National Cancer Centre Singapore and Duke NUS Medical School set out to do something that has never been done before: map the entire genetic genome of the durian. The task proved to be a Herculean effort, as 45,335 protein encoding genes were identified. Compare that to humans with 20,412, domestic dogs with around 19,000, and durian's closest relatives, cotton and cacao, with 41,330 and 29,071 protein encoding genes respectively. The study also found that the distinctive aroma of durian is comprised of at least two groups of volatile compounds. The first being volatile sulfur compounds, or VSCs, which include things such as thiols, disulfides, and trisulfides, which the team hypothesized may contribute to the roasted, sulfury, onion-like odor that is so distinctive of durian. The second group of note is that of esters, which was associated with the sweeter, fruitier odor some experience when smelling durian. While analyzing the genome, the research team made an interesting discovery to do with an enzyme called methionine gamma lyase, or MGL for short. They found that there had been an evolutionary expansion of the MGL gene into 4 different copies, which they naturally named MGLA1, MGLA2, MGLB1, and MGLB2. MGLA1 and 2 were shown to share genes with the relatives of durian such as cotton and cacao, and were deemed to be the conserved ancestral MGL gene, while MGLB1 and 2 may have diverged in sequence after a genome duplication and thus becoming novel to durian. Other than MGL, the team observed significant genomic expansions at the pathway level in gene families related to sulfur metabolism. The gene families the team identified included ATP sulfurylase, evolved in the biosynthesis of hydrogen sulfide, OASA1, which maintains sulfur levels, THIK, which produce lipid volatiles, XBAT32, a regulator in ethylene biosynthesis, ETR2 and AHK5, which are involved in ethylene signaling. The research team posited that the gene expression of these families being upregulated in durian may be the key link to the increased VSC production of the fruit and a major contributor to its odor. Now, what makes this so important? Well, as detailed in this study by Fabrice et al., studies and plants have shown that MGL is a major functional contributor to VSC biosynthesis, degrading the sulfur-containing amino acids cysteine and methionine into methane thiol, which is further broken down into di and trisulfides. And, as touched upon before, di and trisulfides are known for producing a sulfury roasted onion-like aroma. Now imagine this aroma cranked up by 2000% due to the consistent upregulation and expression of MGLB and especially MGLB2 in durian during its ripening. You can understand why people find the smell so offensive. Ripening is of course part and parcel for any fruit. However, there are 2 distinguishing categories that fruits fall into while ripening. The first being climacteric fruits, whose ripening is traditionally seen after being picked. Fruits such as apples, bananas, and durian are popular examples of these fruits. The second being non-climacteric fruits, whose ripening does not continue after being picked. Fruits such as strawberries, pineapple, and watermelon fall into this category. What is it about climacteric fruits that make their ripening process continue even after being picked? It's their relationship with ethylene gas. Climacteric fruit, after dropping from the plant or being picked, produce higher amounts of ethylene than that of their counterpart. This relationship between elevated ethylene levels, fruit ripening, and durian odor seems to point to the association between durian's peculiar genome and an essential amino acid by the name of methionine. Methionine is the precursor amino acid for both VSCs via MgL and ethylene via a key ethylene production enzyme involved in ripening called ACS. With the genomic expression of both MgL and ACS being upregulated in durian fruit, this suggests that over the ripening period, as MgL and ACS expression is going into overdrive, so are the levels of both VSCs and ethylene. So as the levels of VSCs and ethylene increase over the ripening period, so does the odor properties so commonly associated with durian. Another research team from the German Research Center for Food Chemistry decided to take a different route in discovering what causes the durian aroma. Their plan was to systematically analyze individual durian volatiles and assess their contribution to the odor of durian. They did this by isolating durian volatiles and creating a solvent-assisted flavor evaporation distillate from durian pulp. The team then had 6 trained panelists smell the aroma of each compound to see if they could perceive it. This continued, but with each subsequent smell, the compound was continually diluted until the panelists could not perceive the aroma. This gave the research team something called a flavor dilution factor, or better known as an FD factor. To put it simply, the more the compound had to be diluted to eliminate the aroma, the higher the FD factor score. What was found was that a fruity odorant by the name of ethyl-2s2 methylbutynate had the highest FD factor score with a massive 16,384. This dwarfed the second place ethyl cinnamate with its honey-like odor and FD factor of 4,096. And third place 1-ethylsulfonol ethanethiol and its roasted onion odor and FD factor of 1024. Interesting enough, of the top 13 most aroma active compounds found in durian, fruity and roasted onion odorants feature most heavily with 3 apiece, followed by caramel with 2, and honey, sulfury onion, soup seasoning, skunky, and sulfury durian each with a solitary entry. This combination of the extraordinary high FD factor of the sweet-smelling Esther ethyl-2s2 methylbutynate mixed with the various pungent-smelling sulfur compounds and sweet esters of varying odor and intensity give off the complicated rotten but sweet smell of the durian. The bizarre yet visceral relationship humans have with the durian and its ever so fascinating smell while ripening can very well be linked to us as primates and our advanced olfactory system. This shift in odor while ripening is commonly seen in plants whose main dispersal vectors are primates who are enticed by odor instead of other visually enticed animals. Whether you're a fan or an adversary for durian, you have to admit there is something special about it. The strong smell that sees it banned from subways, hotels, and even some public places entirely is kept safe through the love of its supporters, the impact to the economy, and through the curiosity of the world over.